Got it. Okay. How do I present? Cool. Okay, I'm gonna talk briefly about CDK4/6 inhibitor for breast cancer. So there's different types of breast cancer categorized according to like what kind of receptor is found on the breast cancer cells. There's HR positive, which means that hormone receptors can be found on the breast cancer cells, and this is the most like commonly found breast cancer. And there's also HER2 positive, and there's also triple negative, which means like all three kinds of receptors cannot be found on the breast cancer cells. And this is like the type of cancer that's the most hard. That's like the hardest to find the treatment for. And the treatment I'm talking about today is an inhibitor. And the commercialized drugs in the market so far is Pablo. Yeah, I'm not gonna try to pronounce them. But basically how it works is that CDK, also known as cyclin-dependent kinases, as you can tell from the name, it needs to bind to cyclin in order to work. Excuse me. So basically it regulates cell cycle entry, and other factors that play into this mechanism is RB protein and E2F transcriptional factor. So basically, RB protein is bound to the E2F transcriptional factor, and by doing that, it regulates the cell cycle by inhibiting this factor. And what this factor does is that it provides important expression of genes for DNA replication and cell division. So when the RB protein is bound to this transcriptional factor, the cell cannot enter the S phase from the G1 phase. So that's what it does. But oh my God, my voice. What this CDK thing does is that it phosphorylates the RB protein, and when that is done, the RB protein releases the transcription factor, by which it activates it, so the S phase can begin. So for this treatment, yeah, so the mechanism basically works by inhibiting CDK 4 and 6, which are in charge of activating E2F transcription factors. Yeah. So what I want to talk about mainly is the resistant mechanism about CDK4/6 inhibitor. So for the ER positive, like the hormone positive breast cancer, the main treatment so far is ET, which is endocrine therapy targeting the hormone part of the breast cancer, and CDK4/6 inhibitor. And why we're using a combination of the two is because both have shown resistance. Basically, persister cells develop, which is why like a combo might work better. And so far, research on CDK4/6 inhibitor resistant mechanism has been pretty lacking. And the main mechanism that research so far have found is the loss of functional RB. Basically, like mutations have happened in RB protein, so that no matter how much CDK4/6 inhibitor you put in a patient's body, the cell cycle continues on because basically there's no RB protein, and therefore the inhibitor can't work through the inhibiting mechanism. So current research has basically Oh, 我忘了 start time. Okay, speaking about like okay, 我也快讲完。But yeah, the current research has so far moved on to like non non non-genome genome oh my god, I'm trying to non-genomic mutation. So basically that mutations that occur when the cells require it. Because so far, even though researchers have discovered that there's like this loss of functional RB, but still like after for the ER positive cancer patients, breast cancer patients, there's they don't develop this somatic mutation like after moving into the therapy, and they found out that low levels of E2F activity is still present. So the question now is like if there's any other, for example, like signaling pathway or growth factors that play into this resistance mechanism. And we are also trying to figure out if the use of CDK4/6 inhibitor can extend to other subtypes of breast cancer, for example, the triple negative breast cancer, because so far it's being proven to be pretty resistant in triple negative, and there's not like good therapy for triple negative in the market so far. So yeah, that's like where the research is guided to at this stage. Yeah, that's basically my share.
Is there any like particular reason why you think this confident?
Oh, it's my lab is doing this, so yeah, I did some research on this. I found this pretty interesting. Also, there's like a lot of medical burden with breast cancer, so I think yeah.
What's the overall treatment landscape of breast cancer? 我知道 HER2 好像是有特效药是吗？HER2 positive 还是就是 like 不同 subtype？
对，它就是 breast cancer，它 treatment 就会非常的 specialize，就是非常 individualize，因为不同的 individual 会有不同的 resistance，然后你就需要，比如说荷尔蒙的，就可能需要 ET 和这个 CDK 在一起，然后 HER2 我不是很了解，然后 triple negative 它可能就是更多的是像 chemotherapy 或者直接切掉这样子。
CDK 那个图是你自己画的吗？
Academy。Yeah。
就是想说，课把我还没学到这儿吧？
没学到，没学到起的差。
那个。
Okay, I'm gonna share screen. 我有套给谁看？ Are you looking at my G cal? Anyways, okay, 我 start timer，不是这个，大家看得见吗？OK， 今天给大家讲的是 Fanzor 这一套，就是 programmable RNA guided endonuclease. Before I start, I want to give like a quick overview of what CRISPR is. 大家 like probably knows, but I just thought it's like a cute little graph made by Introbio, Dr. Adner. So basically, I thought we always forgot that the original CRISPR system worked by, for example, it first incorporated different viral DNA sequences into its own kind of like genome or bacterial genome. And then these CR or CRISPR RNAs get transcribed into like actual RNAs. And these RNA serves as like a guide RNA for the CRISPR-Cas system that binds to the target DNA when the viral infection, when like virus enters the bacteria. So essentially, it's an RNA-guided endonuclease system, and that is what really matters. And 之前这一套就是这些CRISPR 和OK，之前这些 CRISPR 的话呢，都是在bacteria 里边发现的，在 prokaryotes 发现的。然后在差不多1年前还是半年前左右，反正张峰那个团队呢发现了一套就是类似 CRISPR，it's actually CRISPR 之前的一套或者说是 CRISPR 的 ancestor 的一套系统，叫做 Omega。然后这里边有个 protein 叫 TMPB， that basically perform the similar function of like cleaving the like target DNA。 然后这个图呢基本上讲的就是说Omega 这一套 system 它的主要的作用呢是叫做 transposition. Basically what transposition means is it is to like move the DNA around, right? 就是可能它想either it's like the same strand of DNA or like a different strand of DNA they want to incorporate into some genomes. So it's called like transposition. And then the CRISPR system, CRISPR-Cas12, which is derived from this TMPB Omega system, serves as like adaptive immunity. 它主要是 target the viral genome， 对吧？就如果你见到了一个 virus， 然后它会把这个 incorporate 到自己的 genome， 然后帮助你下一次再有 viral infection 的时候，你可以去预防这个。然后张峰团队呢，他现在找的这一套 Fanzor 这套系统 ，Why it's so cool? It's the first time they discover anything in eukaryotes。所以他们之前一直在找在 eukaryotes 里边找这类似 CRISPR 的这种 RNA-guided endonuclease system， 他们都用的是就是 CRISPR-Cas 的这个 Cas12 的 genome 去as a seed to find like similar kind of things， 但是他们就一直没成功，然后是找到了 TMPB 这个更早之前的 ancestor 才是作为了一个 correct seed to help them find this Fanzor protein, right? 然后I think it's so cool that they don't know what this Fanzor is doing in eukaryotes. The biological role is still unknown. They have some guesses, but it's all not like proven. 就是他们还没有去做，比如说 knockout 这个会产生什么效果。This has not been done. But yeah, so I guess this first figure 他们就是这一套 basically shows like 就是Fanzor 1和 Fanzor 2都是从 TMPB 去 derive 的吧。然后 Fanzor 1是it's 在这两个 bacteria 不是 bacteria 应该是guessing they're fungi, they're eukaryotes, right? Multicellular organisms, some sorts of multicellular organism. 然后 Fanzor 2呢，这个是一个 it's a marine organism, so it's kind of cool. Yeah. 然后let me see. Yeah, I guess they let me see. Yeah, so Fanzor 1 and 2 both emerge from like horizontal gene transfer that involves TMPBE gene， 就是说它们基本都是从 TMPBE gene derived 的。然后I guess something really interesting is here 它们都有一些类似的 conserved sites or like there's some similarity between their catalytic site. For example, this NUC nuclease site, REC recognition site. Yeah, and in this graph, so the next experiment they did is to kind of see if these Fanzor proteins actually performs like cleavage. And what they did is they kind of like identify the Fanzor sequence, and they put in like a guide or they put in like a TAM sequence or like a target basically 就是帮助他们去放了这个，所以它可以 target 到他们想 target 的 plasmid， 然后看它这个 plasmid 是不是有被 cut。然后in figure B I think it shows that 就是不同的这些 Fanzor in different organism have different like cutting patterns. They have distinct cutting patterns. 大家可以看到这个地方，比如说它还 generate 了 overhangs， 然后这些地方可能是更直接的，就是把它 cut 掉。然后these figures shows that like mutation in one of these sequence 都会去disturb 它的这个 activity 或者 cleavage activity 然后 it also like for example like magnesium, oh my god, magnesium, calcium kind of like support this activity which would make sense because 它们这些生物的生活环境可能就是 naturally 会更多这些成分in a similar way this graph shows like 它们的在不同的温度下spanning from like 4 degrees Celsius to 70 degrees Celsius 都有同样的一个cleavage activity, which corresponds to the like wide range of temperature that these creatures can survive in. And also it's like specific to like double-stranded DNA, right? 就是比如说他们试了 single-strand DNA, double-strand RNA 和 single-strand RNA 都是它没有办法 cleave, 它只能 cleave double-strand DNA. Yeah. 然后yeah, another cool thing is they started trying if this actually if this can be used as to basically like CRISPR to cleave like human genome, so serve as a human genome editor. I think that's super cool. 然后they kind of like found different ways to enhance the activity, but basically 就是说他们其实这一套系统它的这个 cleavage activity 肯定没有 CRISPR 那么强然后它的 like even though they can enhance it the successful rate is still like 18% or something but I guess one thing is 它不会I think it's like 它不会 disturb 到就是the broader or like the other part of the genome, so it's a very 就比较像是比较 safe 的这么一种 tool 吧 I guess but yeah I guess they're still working on improving the efficiency but yeah, and here's a really cool cryo-EM of Fanzor. Yeah, okay, I'll just
为什么他们一直想找一个 eukaryotic genome editor?
Yeah. I guess 就是 from my perspective, right? I don't know if this is what they think, but I think like 就是如果你自己身体里本来就有这种东西了，对吧？那你或者说有一个类似的这样的一个 endonuclease 的 system, then you would rather use your own than like I don't know if that makes sense, but I don't know if they have tried to identify like similar things in humans because 那些 organism eukaryotes, even though they're eukaryotes, they're like 比较原始的 eukaryotes, right? 它们不是很复杂的so I don't know.
所以你觉得就是说这个东西应用前景应该不是说跟 CRISPR 一样把它作为一个in vitro 的一个实验，而是说in vivo 做能让这些去干一些事情。
Sorry, can you repeat your question?
就是说这个东西的应用前景呢，应该来说，如果说我们能在人体里面找到这个 protein，那我们更希望它在人体里面去在一个自然的它本来的这个环境里面去。
Yeah.
是说 introduce 一个新的 prokaryotic DNA， 对吧 ？Do something, probably in vitro.
Yeah, I guess it's still 你可以 still like design like a for example, the guide RNA to actually perform the function, right? So there's still some kinds of like artificial engineering going on, ideally, if you want to use the systems. So
之前刚刚说就是说他们还没有完全确认这个在这些生物里面的作用是什
yeah,
么，
yeah,
然后
yeah, yeah.
他们有一些猜测，大概是什么样的猜测？
他们有一些猜测 That's a good question. I can't remember, but yeah, I guess 就是他们有说，就是说，transposition can be one of the thing，就是和之前的那些和就是 omega system 类似的 transposition 可能是one of the possible mechanism. But yeah.
因为就是我可能我没有在我的 presentation 讲，
Yeah,
但是
yeah,
就
yeah,
是
yeah.
就是那个我刚刚说那些基因，他们猜测有可能是被就是 endogenous retrovirus 所激活的，所以 I was like thinking maybe like you know they're trying to
Damn, yeah,
integrate
yeah,
these
yeah.
virus sequence into our own genome and then use them
Mhm.
to like activate some sort of intrinsic
Yeah, yeah.
Same as CRISPR.
yeah, but like just in stem cells.
I guess, do humans have endonuclease? I guess.
对
但是确实
我
有
在想，
就是
我记得应该是有类似的，对吧？哦，transposons, yeah, yeah, yeah. Yeah, yeah, yeah.
says this place.
为什么它叫 Fanzor？它和是封章有关系。
That I do not know.
真知道。Acronym is FZ。
I don't think it stands for anything. Yeah, 它好像 it's not, it doesn't 就不是 CRISPR， 它是每一个字母 stands for something, right? I don't think this stands for anything. Omega 是 stands for something， 但是 Fanzor 不是。
Fanzor 的 behavior 也是 cuts double strands，
对 ，double strand DNA, yeah.
但是它会有 overhang。
Yeah. It depends on the type, I guess. 对对对。然后它其实挺有意思，是每一个 organism 它们自己的这些还都不太一样，就它 cut 地方还都不太一样，so yeah. OK，那谢谢大家。